Good morning. This is Dr. Raju Devde, Professor and Head of the Department Pharmacology, Kaktiya Medical College, Warangal. Today, the topic is new drug development, clinical trials, and good clinical practice. So, the phase 2 undergraduate MBBS students, by the end of this session, should be able to know why a new drug is required, why the drug analogs or its derivatives or substitutes are developed, what is the approach in new drug discovery or invention, how to develop a new drug, introduction of a new drug, and not only that, they should be able to understand the rational introduction of a new drug to human beings that will be considered under the clinical trials. Apart from that, the phase 2 student should be able to describe the clinical trial and its phases. And finally, they should understand why there was a need for the development of guidelines with respect to good clinical practice. With this objectives, we'll look into the topic. So, in new drug development, which is a process that applies to drugs, products, and protocols to be used on human subjects, Then, if you look at the new drug development, which is divided into 4 phases. Number 1, the drug discovery, the preclinical development, the clinical trials, and the approval of the drugs, which is called as post-approval phase. Now, what are the goals and objectives of the new drug development? The goals and objectives are the clinical pharmacology and pharmacometrics. One should know assess the safety of the drug, the activity of the compound or the drug to be tested, its effectiveness, what differentiation does it have with the previous drug which was there in the market, what advantage that it has over the older version of the drug, then followed by successful FDA submission of the data which are extrapolated from both the preclinical as well as the clinical trial to the FDA, that is US FDA, United States Food and Drug Administration, US FDA. Then finally, after the drug is approved, after submission it is approved by the FDA, the drug will be expanded in the market, that is market expansion followed by post-marketing surveillance. That means to say, the molecule or the compound which is tested has to be vigorously screened, that is the preclinical testing as well as the clinical testing. And once it gets cleared from all the various stages, when it comes into the market for its use at large in the population, we need to do a surveillance of this particular drug. Why we require surveillance is because the preclinical findings, that is the testing which is done in the animals, the effects, the safety, the ADRs totally cannot be extrapolated in the humans. And also there are various limitations for the clinical trials which occur in the human beings. That is, the duration of testing is limited. Then certain subset of population is recruited for the testing of the drug. And not only that, pregnancy, lactation, children, old people are not included in the disease or to be tested, the drug which is tested upon a particular disease. So these are various limitations. That is the reason why once the drug is expanded or when the drug is manufactured and released into the market after approval, we need to go for a post-marketing surveillance, which is called as pharmacovigilance. Now, if you look at this particular FDA concept of the 3 dimensions of the critical pathway in drug development. So if you look at this particular thing, we have a basic research here. This basic research, various dimensions which are tested in basic research. For example, basic research, there is safety, medical utility, and industrialization. So this safety, medical utility, and industrialization, basic research is followed by prototype drug design or discovery, which is followed by the preclinical development, which is followed by the preclinical development. And not only this preclinical development, the clinical development, and then we have the FDA filing and approval and launch preparation. Okay, so these are the various stages. For example, basic research, then we have a prototype design and discovery, followed by preclinical testing, then followed by clinical development, followed by FDA filing and approval. Okay, followed by FDA filing and approval. Then when you look at the safety in the prototype design, if you find a safety in the prototype design, there's a material selection, which is nothing but studying the structure activity relationship. In the preclinical testing, there is in vitro and animal testing. In the clinical development, if you look at the safety, the human and animal testing occurs, which is followed by, again, the safety follow-up. If you look at the prototype design and discovery in medical utility, again, there is an in vitro and computer module evaluation which occurs. And not only that, followed by in vitro and animal models which are tested. Then we have human efficacy evaluation, the medical utility, which comes especially in the second and third phase of clinical trials. Then if you look at the last one, that is industrialization, which is the prototype design, we develop a physical design, which is followed by characterization and small scale production of that particular drug. Then once there is an approval, there will be manufacturing or scaling up of refined specification of the drugs, that is followed by mass production of the drug. So these are especially what you call like the FDA critical path in the drug development. Okay. Now, when you look at the next aspect, sorry, okay, right? When you look at the next aspect, is this a costly affair? Which one? The drug development process or the preclinical testing or the clinical? Yes, definitely. It is very much costly. And not only it is very costly, but there are some thousands of molecules or the compounds which will answer the following question in medical research. But out of those thousands of compounds, 99% of the compounds are a failure at a given particular stage. It could be a preclinical testing, or it could be clinical testing, that is the clinical trials like phase 0, phase 1, phase 2, phase 3. Okay, so they may fail at any point of time. And this particular aspect of new drug development is very costly. It may cost some millions of dollars, and it is a lengthy process. At least a minimum of 12 years to 15 years are required for a drug to come into the market, So that we have to remember. And any compound, it can fail at any given stage of the clinical testing of a drug. So that is very important. So it is a costlier effect. So that is why we say evidence-based medicine. Now, ours is not an allopathic medicine. It is what you call like modern medicine. So it is all evidence-based. Now, if you look at the drug discovery process, what happens here? Drug discovery process generally occurs due to screening, which consists of testing of many compounds in various assays relevant to the disease in question. So this is what is called as a HIT. So what is a HIT? HIT is nothing but screening consisting of testing many compounds in assays relevant to the disease in question, which is called as a HIT. And if the compound or its structural derivatives continue to show promise after further biological and chemical characters, which is called as lead. So there are some thousands of hits which are tested, out of which we get some hundreds of leads, which we will be tested further. They go ahead with the clinical trials, and out of those hundreds of leads, only a few molecules or only a few compounds, they will satisfy all this vigorous screening procedure and are released into the market. Okay, so if one has to remember that whatever the drugs which are available in the market for clinical use are very stringently processed, and the procedure of screening of these drugs may take around 12 to 15 years. So there are thousands of leads, sometimes more, and some thousands of hits, and some hundreds of leads, out of which a few will be successful in clinical trials. Now, what are the basic strategies which are applied in new drug development? There are two basic strategies. One is the compound-centered strategy. What is that? The compound-centered strategy. In the compound-centered strategy, the second one is what is called as the target-centered strategy. When you talk regarding this compound-centered strategy, like the natural products which are isolated from the plants, which are isolated from the animals or the microorganisms. So they are natural products. Not only that, the few examples of these natural products which are there in the market, like morphine, quinine, atropine, adrenaline, thyroxine, and penicillin. Of course, everybody knows these are either as penicillins or the plant sources like morphine, quinine, atropine. They are the plant sources like that. Okay, so this is how we can say that yeah, the nature, that is the compound centered approach. Next, there are certain natural ligands. So what are these natural ligands? So natural ligands are nothing but there are certain diseases. For example, Parkinson's disease or Parkinsonism, which is also called as paralysis agitans. If you look at this Parkinson's disease, the pathology in the Parkinson's diseases, there's a deficiency of dopamine. So this deficiency of dopamine can be treated with levodopa, L-dopa. Then when you look at diabetes, especially per se if it is type 1 diabetes, which is called as insulin-dependent diabetes mellitus, there's a deficiency of insulin for which we give exogenous insulin like that. So the drugs can be developed in such a fashion. If you look at the target-centered approach, in the target-centered approach, we use biochemical or molecular targets, and they have certain advantages. For example, when you target a drug, either the biochemical or the molecular target, the advantages are we can test for the pharmacological activity, and also this can be tested in the systematic assays, or the system assays can be conducted in testing the biochemical and molecular targets of that particular compound under evaluation. Not only that, there are certain structure-based approaches. So these structure-based approaches, they use the 3D structure of the target obtained through X-ray crystallography or nuclear magnetic resonance. Okay, so the structure-based approach in the drug development utilizes the 3D structure of the target, which is obtained through either an X-ray crystallography or nuclear magnetic resonance. The advantage of the structure-based approach is to know the potency of the drug, and in this particular aspect, limited number of drugs can be tested. The disadvantage is the synthesis of this particular compound is a problem. In the 3D structure, the synthesizing will be a problem. Not only that, if you look at the chemical synthesis, so this chemical synthesis of drugs are based on the structure activity relationships. So there are so many examples wherein you can just modify the structure of a compound to get some advantage over the previous compound. If you look at the structure activity relationship, so just a slight modification of H1 or histamine, okay, in the structure of the histamine H1, the histamine blockers or H2 receptor blockers are derived. A slight modification in the structure. Then there are certain enantiomers. So what are these enantiomers? They are nothing but either a levo form or a dextro form, or we call isomers of that particular compound. So these isomers, when they are available in different forms, for example, there are atenolol. So the stereoisomer of atenolol is S-atenolol or S-metoprolol. So this atenolol and metoprolol or S-amlodipine. So atenolol and metoprolol are beta blockers. S-amlodipine and amlodipine, it is a calcium channel blocker. So if you look at this atenolol, metoprolol, and amlodipine, when compared to S-atenolol, S-metoprolol, and S-amlodipine, the potency is increased to twice when compared to the normal forms like atenolol, metoprolol, or amlodipine, right? Like that, we have cetirizine to give an example, which is an antihistaminic drug. There is a loratadine, ketoprofen. So cetirizine, loratadine, they are antihistamines. But when it is converted or when it is changed to enantiomeric forms like isomeric forms like levocetirizine, deslevocetirizine, or dexketoprofen, their potency is increased to or it is doubled. Like that, we have another drug which is called as atracurium and cis-atracurium. So atracurium compared to cis-atracurium, the potency of atracurium is 1/4 when compared to that of cis-atracurium. That means cis-atracurium is 4 times more potent. So this is an advantage of a chemical synthesis of a compound based on structure activity relationship. And not only that, in the structure activity relationship, so a drug can be made longer acting also. For example, lansoprazole is there. So lansoprazole, which has a different duration of action in peak effect. Okay. But when you compare to that, the lansoprazole, which is a proton pump inhibitor, to dexlansoprazole, it has a longer peak effect when compared to that of lansoprazole. Like that, we can give so many examples. So many examples. Or for example, we can change a compound or a drug uh, based on the structure activity relationship by slightly modifying the structure. We can change the or we can alter the pharmacokinetics. For example, we have a drug which is called as omeprazole. So this omeprazole is a proton pump inhibitor, and then we have S-omeprazole. So this S-omeprazole is having better oral bioavailability. Like that, we have ofloxacin and levofloxacin. So this levofloxacin is having a slower excretion through the body. Hence, it has a better action like that. So this modifying a simple structure, slightly if you are modifying the structure, we can increase the potency. Not in that, we can increase the peak effect to longer duration, and we can increase the pharmacokinetics like the better oral bioavailability, slower excretion, less adverse effects, and so on and so forth. Then we have another thing which is called as rational approach, like we are targeting the proton pumps and reducing the acid secretion, especially in the acid peptic disease. Then we have drugs which are targeting the molecular level, that is molecular modeling of the structure, where we have COX-2 inhibitors, like selective COX-2 inhibitors, preferential COX-2 inhibitors, which are actually, they reduce the pain. So generally we have non-selective cyclooxygenase inhibitors, like aspirin. We have preferential COX-2 inhibitors like nimesulide. We have selective COX-2 inhibitors like rofecoxib, atorvastatin. So these drugs, they have better profile when compared to the adverse drug reactions. They are gastroprotective COX-2 inhibitors. When you look at the combinatorial chemistry using biotechnology, where we have various growth factors, hemopoietic growth factors, cytokines which are there into the market, which are used therapeutically like that. Then what do you mean by lead optimization? So I told you regarding hits and leads. So hits are compounds which are say thousands of compounds which are targeted towards a research question, and out of which we get a few leads. Okay, so what is this lead optimization? So lead optimization is the stage where the physical, chemical, biological, and pharmacological properties are characterized and refined with the ultimate goal of selecting a single molecule to enter into the clinical testing and formal drug development. That means to say, it is just like an inverted pyramid. To start with, there are some thousands or even lakhs of hits. So when it comes to leads, it is tapering down to some few thousands. But optimization is something in the new drug development where this stage where the physical, chemical, biological, and pharmacological properties are characterized and refined with the ultimate goal of selecting a single molecule to enter into the clinical testing, and formal drug development will occur, and then it is subjected for approval with the what you call like the FDA, and the drug is marketed for use in the general population at large. That is lead optimization. Now, when you talk about drug discovery, so before going through all these things, so drug discovery most of the times is serendipitous. So what is this serendipitous? Serendipitous type of drug discovery is something where the drug is discovered by chance. So there are several examples I can quote where the drug is developed or investigated for some purpose in a planned way, and then it has been found that that particular drug has some other significant effect. And on further thorough investigation, a clinically significant effect will be noticed. For example, the penicillin G, discovery of penicillin G, which is an antibiotic you very well know. This is discovered and it has a very a good role to play, has played in the Second World War. Not only that, the drug discovered through serendipity was phenytoin. It is an anticonvulsant. Then anti-diabetic drug like the pioglitazone, which is a thiazolidinedione, which was also discovered through chance. Such drugs will be approved for clinical use. And there are certain discoveries of the drugs which were made by, or the drugs were discovered by medical students, which are of very highly clinical significance. For example, one medical student called Paul Langerhans in 1869 showed that pancreas has two types of cells. First one was related with digestion, the other was with different function. There is a person called, a student called Sandstrom in 1819 discovered human parathyroid gland. Then another medical student I can quote here is J. MacLean in 1916 discovered heparin, and Bantin was a general surgeon, and his student was Best in 1922 discovered insulin. So these are various breakthroughs or in the research and development or the new drug development. So one has to be very keen that serendipity or by chance discovery is also possible. We are having so many examples for that. Apart from that, this drug discovery identifies interesting drug compounds, drug targets, and delivery mechanisms with the potential for development into products. And these products, which characterize compounds and their targets, are proof of concept in nature. And they provide fundamental information on target biological system and to identify the lead compounds for further development. So it is a process. Drug discovery is a process which characterizes the drug compounds, drug targets, the delivery mechanisms, the potential of development into products, which characterize the compound and the targets, and finally, the proof of the concept which is implemented in the nature, provide fundamental information on target biological system, identify lead compounds for further development. Now, after this, there is something which is called as preclinical drug development. So preclinical is something which is before the human trials. That is something which is dealt with animal studies. For example, this preclinical drug development spaces the gap between the drug discovery and the clinical trials. They also provide data on the safety and efficacy of the product to be tested or the compound to be tested. And these include in vitro studies. So in vitro is something, for example, you do some bioassays or you do some testing in vitro. In vitro is outside the body, or you take a tissue and you test the compound there, or you take cell lines and you test the compound there. Okay, so it includes the in vitro studies, drug manufacturing, formulation and packaging, and also in vivo studies where you take a whole animal and do a study. Okay, so in vitro and in vivo. So if you look at in vitro study, as I already told you earlier, the intact cell lines and cell-free system. So what are these intact cell lines? So these intact cell lines, they assess the drug effects on the cells with specific DNA mutations. Cell-free system is nothing but assay of the enzyme activity, receptor binding techniques, the protein interaction with signal transduction in vitro studies. Then when we look at drug manufacturing and formulation, so this drug manufacturing and formulation are conducted under current good manufacturing practices guidelines. So if the manufacturing practices are not correct, or we don't have stringent guidelines for manufacturing of the drugs, what will happen? So many spurious companies will come up. Of course, there are so many spurious compounds, so many spurious companies which manufacture with a substandard molecules, or their binding agents, excipients may differ. Okay, so the manufacturing and formulation of a compound, once it is approved, it has to be conducted under a good manufacturing practices guidelines. So we are having certain governmental regulations or the ISO standards. So ISO certified manufacturing units or the research and development units should be there. So if you look at the in vivo studies, we have the pharmacokinetic studies, which deal with absorption, distribution, metabolism, and elimination and excretion of the drug studies. Then we have PD or pharmacodynamic studies, where we exactly assess how the drug is going to act, what is the mechanism of action of these particular compounds which are being tested. Then the therapeutic index is very important. So this therapeutic index is very important, which is dealt especially with the LD50 by ED50, and the compounds which have the narrow therapeutic index have very toxic effects, so on and so forth. So we also follow with this toxicological studies. And these toxicological studies, generally, there are certain objectives set, and these toxicological studies will be acute, subacute, chronic studies, the teratogenic studies, the mutagenicity studies, which are again the very, very time-consuming studies. And it has certain method to be followed, and the aim is to provide the safety of the drug. Toxicological studies, the acute toxicity study, where you provide the safety of the drug and you assess what is its therapeutic index in the observation. There's something which is called a subacute study. And in this subacute study, again, it could be some short duration study to identify the target organ, which may result in a damage, to determine the clinical parameters, to determine or to estimate the safety margin of that particular compound. It also has certain methodology and evaluation, of course, that will be dealt with in detail in later classes. Then apart from that, we have a chronic study. It is similar to subacute study, except for the duration, which is quite longer when compared to subacute studies. Then we have certain studies which are called as special studies, where we are going to test the effect of the compound on the reproductive system, and whether it's having any teratogenicity, the mutagenicity, and carcinogenicity. Again, they are, once the drug comes into the market, these particular effects can be tested upon. It's again a long-term study, which is a special study. Then after all these particular screening, the clinical drug evaluation authorization will occur. That means to say, once the drug is cleared of this preclinical testing, okay, then it has cleared with certain positive aspects and with less of the side effects or adverse drug reactions. That is, the safety of the drug and the efficacy is tested in the animals. Then the drug, the compound, we go for an IND, that investigational new drug. So what is this investigational new drug submission? So once it is cleared of preclinical testing, we submit the data to the FDA for its investigation in human beings. So the rationale for the drug and the patient group to be tested. Okay, so this investigational new drug submission consists of the rationale for the drug and the patient group to be tested. And all preclinical safety and efficacy data are pulled up, and a detailed plan for clinical development or clinical investigators brochure is released, and it is submitted to the FDA for review and permission to proceed in the human beings. So this is the first time the compound cleared the preclinical testing with the positives, that's a good efficacy and good safety data. So safety is good. It is very efficacious in the what you call like the animal studies or the or in vitro or in vivo animal studies. Then it is submitted to the FDA for review and permission to proceed for its testing in the human beings. For this testing in the human beings, there are certain ethics to be followed. So these ethics are very important. Why? Because earlier what used to happen, that is before the World War and even before that, so the drugs used to be delivered in the market with various side effects. For example, there are certain areas like after what you call like this thalidomide tragedy and sulfanilamide tragedy. So it has come out with various ethical regulations. The one which is called as the famous one, which is called as the Belmont Report and Declaration of Helsinki. So this Declaration of Helsinki and Belmont Report, it mainly formulate certain guidelines or ethical guidelines for conducting research with respect to the physician as well as with respect to the patient or the person who is under investigation. For example, the clinical trial must minimize the risk for the participants, and it should give provision for the care of the patients, and it is associated with termination of the clinical trial when the risk becomes incompatible with the goals of the trial. That means to say, we have set a research question that this particular compound is going to reduce the pain in the patient with osteoarthritis. But if the drug or the compound under investigation in a clinical trial is causing cardiac arrhythmia, may cause a fatal cardiac arrhythmia. So we have to stratify the risk and benefits. So risk benefit stratification with that particular compound under investigation is having more of risk and causing serious morbidity or mortality in the person under a trial, that particular trial can be terminated. And not only that, adverse events to be reported immediately to ethical committee. So that is regarding the ethics. And this ethical committee also reviews the protocol before the study is allowed to start. And their job is to ensure that the risks of being in the study are not greater than the potential benefit. So the risks should be minimal than when compared to that of the potential benefits of that particular compound. Okay, then there is something which is entity which is called as IRB or IEC, Institutional Review Board and Institutional Ethical Committee. So what are the functions of these? The Institutional Review Board, IRB and IEC, they act to ensure the rights and welfare of the participant, and the FDA regulations mandates to review the clinical trial protocols for ethical and legal issues. Not only that, also has the authority to approve, modify or disapprove the particular clinical trial. Then there is something which is called as informed consent. What is this informed consent? So a person or the people who are recruited or the participants who are recruited in the study, especially in the clinical trials, okay, they should be always voluntary. The participants should come wholeheartedly and voluntarily getting recruited in the clinical trials. And not only that, they have to be informed what is the procedure which is being conducted, what sometimes they may not know what the drugs are given, but the patient, the participant should be in a position to know what could be the probable outcome, what could be the probable adverse drug reactions, and the participant should voluntarily get enrolled in the clinical trials. And not only that, he can as well voluntarily leave the clinical trial on his own. Okay, and this informed consent has to be taken, written consent, video recorded consent, or the informed consent should be in the local language. So there are so many languages. The multilinguistic, our country is multilinguistic. So the patient should understand in his own language what is to be done, what is being done to him, what are the expected things, what are the areas, how he can be, or whether to continue or not, it is voluntarily can be done. So here, if you look at this particular two pics here, so no, thank you, I'd rather not participate in the clinical trial. Yes, I would like to participate in the clinical trial. So that is voluntary. So the informed consent to again elaborate the purpose of the informed consent. What is the purpose? Why we have to give an informed consent? So what is the purpose of the study? Then what is the medicines to be studied? What are the procedures and the schedules of this particular treatment? What are the risks of that particular study? What are the potential benefits, alternatives to the participation, and the confidentiality? So all these things have to be discussed to the participants in informed consent. So now, after knowing all these things, the ethics, the Belmont report, the Helsinki Declaration, then what are the consent, informed consent. Now, what is a clinical trial? So a clinical trial is to identify a health question. So what is the health question? For example, I have one drug, okay? I have one drug for a particular disease, but that drug is having various adverse drug reactions. So if I get one more compound modifying the structure or whatever it may be, framing a question stating that this particular drug is also going to act on that particular disease, with minimal or lesser side effects. So that particular, the prototype drug as well as the drug under investigation or the compound under investigation has to be formulated in a health question. And not only that, after the health question is formulated, we need to develop a plan as to how we have to proceed with the protocol. Then followed by enrollment of volunteers and the follow the plan. So generally these volunteers, here we are talking about volunteers. In phase 1, we recruit healthy volunteers. Phase 1, phase 2, again we recruit the patients. Okay, like that. So we'll come to that in the later part of my talk. Apart from that, the study, the information collected. So after recruiting the volunteers and follow the plan of action with respect to the clinical trial, study the information collected, share the results with others, and improve the treatment. So this is what has been a flow chart. That is, what is a clinical trial? First thing is we have to identify a question, develop an action plan, enroll the volunteers, follow the plan, study the information collected with respect to that particular study, and share the results with others, improve the treatment. So net thing generally when you look at is nothing but, what is research actually? Research is nothing but, there is something available. I am doing research in modifying that and bringing out a newer product or a drug which is more beneficial to the human being at large or more beneficial to the population at large with minimal side effects or minimal adverse drug reactions. So more safety and less, it should be more safety and more potent with less adverse effects. Now, the historical minute, the first clinical trials, when you look at clinical trials, have a long history, even if not acknowledged as clinical trials. Here, if you look at the formal record of clinical trial dates back to the times of the trialists. For example, Dr. Van Helmont's proposal for a therapeutic trial of bloodletting for fevers. You know what is bloodletting? Bloodletting is what is called as phlebotomy. So this phlebotomy or bloodletting was undertaken way back in 1628. At that time, there were no clinical trials. So that is a historical aspect of clinical trials. And Dr. Van Helmont, his proposal for the therapeutic trial of bloodletting, it's called phlebotomy for fevers in 1628. And Dr. Lind, a surgeon who was there in the ship or a big ship, we used to go for voyages. He has done, because people who were sailing in the sea, okay, they were in the sea for months together, sometimes even years or even months together, where they never had this vitamin C available at that time. There were no fruits. So he has done a trial because Dr. Lind, what he has shown is the people who were there as sailors or the people on the ship, they have developed a disease called as scurvy. And he has done a trial by giving oranges to one set of people and lemons or the limes to one set of people. And he has found that the symptoms of scurvy was reduced by doing this. So this is the Lind's trial, which is a famous trial in the scurvy. And this was done way back in 1747. Not only that, if you look at these clinical trials in a nutshell. So this clinical trials in a nutshell, yeah, the clinical trials in a nutshell, if you look at, I will just, 1 minute, I will just tell you. Yeah. If you look at these clinical trials, First thing is there should be approval of the protocol. The protocol has to be approved in the first aspect. After the protocol is approved, there should be a selection of the investigator. This is very important. Investigator has to be selected. After selecting of the investigator, there is a process which is called as an approval process. After the process is approved, we have to recruit the patients, patient recruitment and participation, followed by data which is filed and registered and obtained. Okay, so here the patient recruitment and participation, where which is followed by data entered and reviewed. Then this not only this data, it is a huge data, it is an enormous data, volumes of material which is being collected, not only in single center, the multiple centers in the phase 3 will come to know, followed by statistical analysis, then followed by presentation and publication of the report, then followed by the data filed and the registration of that product is obtained, and then the product, it will come into the market. It will come into the market. Okay. So if you look at this particular thing, right? So initially, this particular the approval of the protocol, investigator selection, approval process, then patient recruitment and participation, followed by data entered and reviewed. Then we go for statistical analysis, the presentation and publication of the report, and the data is filed and registered. Then registration is obtained, and then the drug, that is the new drug, will come into the market, will come into the market. So this is how this particular drug, the new drug development occurs. Okay, so this is in a nutshell, right? So the clinical trials in a nutshell. So if you look at the screening of a new drug development, that is why it takes very long time. So for preclinical testing, it will take a minimum of 1 to 3 years, and the clinical testing a minimum of 6 to 8 years. So a minimum of 10, 12 or 15 years are required for a molecule or a drug to come into the market for its use at large in the public. So that is the thing. Now, what are the components of the clinical trial? I think, the components of the clinical trial. So in the components of the clinical trial, the one which is called as a review of scientific background, where the hypothesis or the written hypothesis is tested or to be tested. Then we have a study design, a type of the study design, which is a open label, or it is a blind, a single blind, double blind, parallel, prospective study, or a retrospective study, or a comparative study, or the multicentric study, like that. Type of the study design. Then we have a study population, because how much population we have to recruit. What is the population size? Then we have to do a statistical analysis with respect to the different variables and the recruitment of the population, enrollment of the subjects, intervention, follow-up of the subjects, which requires a big, huge organization, and this entire thing which is called as a protocol. Okay. If you look at the phases of the clinical trial, there are various phases. Of course, here they didn't mention phase 0, that is which is called as a microdosing phase. So we have phase 1, phase 2, phase 3, and phase 4. That is, of course, which is called as post-marketing surveillance or pharmacovigilance program. Okay. So in phase 1, generally, we test for the safety of the compound which is tested. So here a minimal of 15 to 30 people are recruited. And mostly here we recruit healthy human volunteers, but there are certain exceptions where uh, 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 we can skip the phase 1, enter into the phase 3. Or for example, in the recent pandemic, like what you call like this COVID-19, so we can even skip phase 2, or phase 2 can be done and they directly go into the phase 3 clinical trial. And there are certain diseases like HIV and AIDS and immune suppress or immune compromised disease, or autoimmune disorders, where generally we don't recruit especially people in phase 1. So phase 1 study where we recruit healthy human volunteers, which consists of 15 to 30 participants. Phase 2, generally we study the safety and efficacy of that particular compound which is tested upon. And here we are going to recruit fewer than 100 people. So here we recruit patients exactly. The patients are being studied in phase 2. Phase 3 generally will look for its effectiveness or efficacy of that particular drug compared to the standard drug of care. And the safety also is tested upon. And here a few hundreds to few thousands of patients are recruited. And this phase 3 clinical trial occurs in multiple centers across the country, or multiple centers across various countries, the phase 3. Then the phase 4 is something, once the drug or the compound is clearing the phase 3 clinical study, clinical trial. So we have discussed now investigational new drug, IND. Then we have NDA, new drug application. So this new drug application, once all these stages or phases are cleared and there is a positive aspect with that particular compound, that compound with all the data, the statistics and the related material is being submitted to the US FDA, which is called as NDA, that is new drug approval. And then the drug will come into the market for its use. And then the phase 4 starts, which is called as post-marketing surveillance or pharmacovigilance. So once the drug has come into the market, it may or may be there for years together. For example, if you see penicillin, it is still there. For you take aspirin, it is still there. You take digoxin, some 5-6 decades, it's still there. Some hundreds of years, the few drugs are there. But there are few drugs which once they come into the market, once they are exposed to the population at large, may show serious signals or serious ADRs, and the drugs are going to get banned in the 4th phase of clinical trials, which is called as post-marketing surveillance or pharmacovigilance. And we in India were doing pharmacovigilance program of India. That is again a new topic that will be discussed in the next class. Now, what is this phase 0? The phase 0 clinical trial, of course, it was not there, but it is a recent designation where included in the US FDA 2006 guidelines, which is done in humans first. And here in phase 0, there is the micro dosing given. That is 1/100th of the pharmacological dose is given to that particular subject. And this is actually not very successful. 1/100th of the dose is given to study the early pharmacokinetic and pharmacodynamic data. And it is having a minimal preclinical study. So drugs which have minimal preclinical study, for example, the drugs which have to be given directly to the people like HIV/AIDS, they go for phase 0, like microdosing, and then look for pharmacokinetic, pharmacodynamic data where minimal preclinical study is required. The advantages are unreliable in vitro and animal studies. If the compound is having unreliable activity in vitro and in animal studies, but the disadvantages is again, because they are not thoroughly tested in animal and preclinical testing, there might be a safety and efficacy issue, which is a disadvantage. When you look at the phase 1 clinical trial, the participants, the dose determinant, as I told you, like a few 15 to 30 participants are there, healthy human volunteers, the dosing. And here it determines the safety of the drug, and it will involve the dose ranging studies to determine the toxicity and the major adverse drug effects. And this phase 1 clinical trial may provide early evidence of efficacy. And the end point of this particular thing is a toxicity of that particular compound. If you look at the phase 2 of clinical trial is for evaluating the efficacy and the therapeutic benefit. And it involves the patients, accounting to 80 to 100 patients. And in this particular phase 2, which identifies the common short-term side effects, and to establish dosing regimen and dose optimization to validate the design of phase 3, because it has to be moved to phase 3. This phase 2 will validate the design of future design of phase 3, and duration of phase 2 commonly it is 1 to 3 years duration, 2 years duration. Then phase 2 again it is divided into phase 2A and phase 2B. So phase 2A is again a pilot study where the dose defining and dose formulations. What is the dosage form and what is the dose defined? Then it also studies the safety and efficacy of the drug with respect to the pharmacokinetic parameters and pharmacodynamic parameters and the data. And then it also studies the risk-benefit ratio in phase 2A. So what is this phase 2B? It is again a controlled pivotal study, which is a placebo, double-blind, which again determines the safety and efficacy of the tested compound. When you look at this phase 3, this is a very important study because here in India now, what is happening is earlier for mild cases of COVID-19 infection with certain criteria which have been involved in mild COVID, where the patient is not having SOB, a shortness of breath, and so on and so forth. SPOT is not less than 94. So earlier they used to give favipiravir, and now there's another drug which is tested, which is becoming a multi-drug or multi-centered trial, which is a phase 3 clinical trial. That is, they are comparing the standard of care treatment in the management of COVID with molnupiravir. Molnupiravir. So this molnupiravir is a tested, is an antiviral drug which is tested, which is compared with the standard of care treatment in mild patients with COVID. So this is the study which is done. There's a phase 3 clinical trial which is done in the multiple centers across India as of now. Okay. Like that, if you look at this phase 3 clinical trial, it is a large multi-center randomized study involving thousands of patients or patient volunteers, not healthy human volunteers, they're the patients. And it is a placebo-controlled blind studies to clearly demonstrate the efficacy, safety, and therapeutic benefit of the compound which is tested. And this particular clinical trial, phase 3, will also have a package insertion and labeling of various adverse drug reactions. And this particular study will extend from 2 to 3 years. Now, what is phase 4? Phase 4 is called as post-marketing surveillance, or it is also called as it is also called as pharmacovigilance. So there is no fixed time and population for this particular study, because this may extend to years, because if you want to know the teratogenicity of a particular drug which is having, or mutagenicity of a drug, or carcinogenicity of a particular drug, it has to be tested for decades to know whether this particular drug, it is having a benefit for this, whether it is going to cause cancer in that person. So there is no fixed time and there is no fixed population. And the purpose is usually to support the marketing campaign and to identify very rarer adverse drug reactions which were not identified in the preclinical testing or phase 1, phase 2, and phase 3 clinical trials. And not only that, to identify the drug interactions between the new compound and other drugs. What are the new drug interactions, and new clinical indication, which is called as phase 5, because you see, methotrexate, it is an anti-cancer drug. It was an anti-cancer drug, but methotrexate is having a new indication. It has been found that methotrexate can also be used in the treatment of rheumatoid arthritis. Methotrexate can also be used in the treatment of psoriasis, like that. So any new clinical indication can also be detected in the 4th phase of clinical trial, that is called as post-marketing surveillance or pharmacovigilance. Okay, now this is again a nutshell. If you look at this is also a nutshell flow diagram where you can see the drug discovery. First aspect, the drug discovery occurs from 2 to 5 years. There are some 50 projects here. The preclinical development occurs where you study the pharmacokinetics, short-term toxicology, formulation, synthesis, and scaling up. Okay, may occur, right? So here, this is called as preclinical development. Then, if you look at the clinical development, again we have this phase 1. Yeah, phase 1, where we actually identify the pharmacokinetics, the tolerability, the side effects in the healthy volunteers. So here you look at whether the drug is safe or not is being tested, especially in what you call like, right? This phase 1, it is meant for pharmacokinetic studies, right? Where you test for tolerability, side effects in healthy volunteers. So here we tested whether it is safe, right? So here there are approximately say 5 compounds here. When you look at the phase 2 study, phase 2 is something where the small scale trials are done in patients to assess the efficacy and dosage of the drug. So here it is long-term toxicological studies are undertaken in this particular aspect. The long-term toxicological studies where especially the efficacy of the drug and the safety of the drug, mostly the pharmacodynamic part is being assessed in the phase 2. So phase 1, whether it is safe, that means to say whether it is having any adverse drug reaction, the safety part, and here the efficacy part, how does it work. Then the phase 3, usually it is a large scale controlled clinical trials where does it work in double blind trials, and here both efficacy and safety are being tested upon. And phase 4, as I told you, it is nothing but the post marketing surveillance. So if you look at these projects in the drug discovery phase, suppose there are 50 projects. When it comes to the preclinical development, only 12 compounds are being recruited here. When it comes to phase 1, only 5 compounds have come. What happened to the 7 compounds? They did not fulfill the criteria. Probably there might be not efficacious, or there might be some side effects. If you look at the phase 2, again, there were 5 compounds, there are 3 compounds. What happened to 2? They might not have met the safety aspect. So when it comes to the phase 3, 1.7 compounds are there. What happened to 1.3? Again, they might not have met the criteria of safety and efficacy. But when it comes to the post-marketing surveillance, the drug, the 1 compound is coming into the market. So this is where this is called as the candidate drug or the drug candidate. That means to say drug discovery is one aspect. When the drug candidate, that is means hits are 50. So lead is say, for example, one no. For example, there are some 12 compounds which are coming as leads. The hits are 50. The leads are say 15 or 20 or 12 compounds. Then these compounds, they come. So if you look at this, it's an inverted pyramid. So out of those 50 projects or some thousands of compounds, 1 or 2 compound is getting approved and has been coming into the market for use in the population at large. Okay, so this is how the particular what you call like the drug development process occurs. Okay. Then we'll go to the next slide. Okay, now I think I will stop my lecture here. I'll continue with my lecture in my next class because I think it is more than 1 hour discussion. So I will stop here.